Hi everyone, in this video we are going to go over three objectives. We will dissect the structure of DNA, we will examine the code within DNA, and using the code from one strand of DNA, we will compose the code on the complementary strand, and we will be able to calculate the percentages of each nitrogenous base within DNA. So here is the structure for DNA, and it is in relationship to the cell itself. And we could examine this in two different ways. We could either go from big, zooming in, like in Google Earth, zooming in to the small, or we could go the opposite way, looking at the small and see how it builds up the big. At this moment, let's go from big to small. So this structure right here, we know it's the nucleus. And within the nucleus, we have these things called chromosomes. Okay, now chromosomes are a full strand of double-sided DNA. So from beginning to end, a piece of DNA is a chromosome. So here's the DNA, and they're showing you that the only way to get DNA to fit into a cell is to really coil it up and pack it tightly. For if you were to unwind all of our DNA in just one cell, every chromosome, it would stand 6 feet tall. All right, so over here, just a portion of DNA, a portion of the chromosome is a gene, and each chromosome has hundreds of genes on it. But getting deeper, what is a gene composed of? Well, it's composed of small subunits that are called nucleotides. Now, just like starches are made of subunits called sugars, just like proteins are built of subunits called amino acids, DNA, deoxyribonucleic acid, is built of subunits called nucleotides. And each nucleotide has something in common, or two pieces in common, but often one piece that is different. You could see the different colors here. Each of these colors represent different nitrogenous bases. So in this case, the red ones are guanine, the blue ones cytosine, the yellow adenine, and the purple thymine. Now this picture right here is very similar. Again, it goes from chromosomes, which are tightly packed strands of DNA, unwinding it. You see a gene here. This time it's not showing you that it's a portion of it, but it is. But what I want you to notice here, in this illustration, the bases are represented by the letters, the A for adenine, the T for thymine, G for guanine, and C for cytosine. And now let's get even more specific right here. So you're going to see what they both have in common. Each one of these, and let me choose that pen, each one of these nucleotides is going to have a phosphate group and a sugar. In this case, the sugar is called deoxyribose. And what helps us understand that it's a sugar is the way it's written. It has an OSE ending. But the thing that can be different are those nitrogenous bases that are on the inside. The outside is called the backbone, and the inside is known as the rungs, similar to a ladder. And that's what this is showing you. Right here, we are seeing the strand of DNA twisted in a spiral called a double helix. But let's unwind it for simplicity. Now, the sides of the ladders are the deoxyribose with phosphate, and they alternate between each other. That's the same for every nucleotide, and it is called the backbone. But the rungs are made of nitrogenous bases. The examples are adenine, thymine, guanine, and cytosine once again. And it's interesting. If you notice a pattern, the A's, the adenines, are always pairing with the thymines on the opposite side, and the guanines are always pairing with the cytosines. Always. If it doesn't, there's a mechanism where the DNA will repair itself. And if it doesn't, well, that could be one reason for a mutation. Now, how does this translate into all these different traits that we have? Well, to understand how a molecule of DNA translates its code into a protein, because ultimately it's proteins that are responsible for our traits, let's see how computers use processors to produce what we see on the screen and even hear. So, how does a computer code translate its information into graphics, video, and more? Well, they do something called a binary code. You see this 1 here and the 0? Binary means 2, so it has the option of 1 or 0. Well, just like a switch, it could flip to the 1 or the 0, and either electricity flows or it doesn't. There are only 2 choices. It's either on or it is off. All right, so how can 2 choices turn into a very complicated processing computer platform? Well, this one's going to take a little while. There it is. So inside a computer chip, there are billions of switches. So instead of just one of those switches, there are billions. So how can this fit into a chip? Well, it's very small. They're microscopic switches. And that's a good analogy for DNA and how it packs so much information. It's microscopic. Even though it can stretch for 6 feet in length from tip to tip, it must be extremely thin. All right, so here's an example of a binary code, but it's using 8 switches. And in this case, if the first 2 are on and the last 6 are off, well, the computer knows that that means it's 192D. Don't ask me more details on that, please. And in this case, if it's on, off, off, it means 168D. That's a binary code for you. Now, this is how they do the alphabet. Okay? It's a 7-bit binary key. So binary code, to be an A, it's on, off, on. So 7 switches relate to the letter A. Now, if you are curious, well, when you look for a computer and you see that thing that says 64-bit, that means that their binary code, or the number of switches, extends for 64 of them. It could handle a much more complicated code than that of just a 7-bit. All right, so imagine if there were thousands of switches. Well, genes are thousands of bases long. So there's an analogy for you. Now imagine if there were more than just options, 1s and 0s, and there are. We have the choice between 4. It can be A, T, C, and G. So again, we're thousands of switches big, and instead of just 1s and 0s, it's now A's, T's, C's, and G's. A very complicated code. So we got to learn about this thing called complementary bases now. And remember, if I went too fast, you could always stop and rewind and redo things. All right, so complementary bases, which are complementary strands, and there's something called Chargaff's rule, and it's just as simple as this. Every time there is a G, a C complements it on the other side, so the other strand. Every time there's an A, there's a T, and vice versa. And it looks something like this. All right, so what I'd like you to do is attempt to, use the single strand of DNA that's given here on the left and compose the order of nitrogenous bases on the complementary strand. I challenge you to pause, try it, and then start again to see if you're right. So, pause. All right, let's see if you were right. Did you get G, A, C, T, G, and T? I sure hope you did. But now, what is missing from this side? And yeah, it is the alternating sugar phosphate. And this just came to me. Let me see if you could figure out how many nucleotides are present on the screen right now. So just like before, pause for a moment, gather your answer, and hit play again. All right, here's your answer. This is 1 nucleotide. We've got 2, 3, 4, 5, 6 on the left-hand side, meaning we have 6 on the right, so a total of 12 nucleotides, which brings us to our conclusion. Now, if 10% of the nitrogenous bases on a double strand of DNA are A, what percentage are T? Pause, think it through. All right, so if we have 10% that are A, well, don't T's complement A's? Which means there also must be 10%. This brings us a total of 20% of A's and T's. So now we have this question. What percentage are C's? That's a little more complicated. All right, for that, we must remember that if 20% are already A and T, that leaves 80% must be G and C. And if for every time we have one G, we have one C, we must take that 80% divided by 2, brings us to 40%. 40% are C's. That means 40% are G's. 10% are T's and 10% are A's, giving us a total of 100% of the nitrogenous bases accounted for. All right, we broke down DNA into its pieces. We built an analogy to a computer so we understand that the code is quite simple. Actually, it seems simple. It's very complex. And then we looked at how the bases complement each other from one strand to another and played around with what appeared to be a very difficult problem. But if you remember that simple rule, that Chargaff's rule, you should be fine. Thanks, everyone.